Asfarviridae, Wikipedia article audio. Asfarviridae is a family of double-stranded viruses. There are two genera in this family. These are the Asfarviridae and the Faustovirus. The Asfarviridae infect insects and swine, and the Faustovirus infects amoebae. The viruses in the Asfarviridae genus infects swine, resulting in an onset of African swine fever. The name of this family and genus are derived from the acronym African swine fever and are related viruses. Only a single species has been described to date for this genus. Asfarviridae. Taxonomy. This group of viruses are the only known arthropod-borne DNA viruses. The viruses in this family are all enveloped and have double-stranded DNA genomes. The Asfarviridae exhibits some similarities in genome structure and replication strategies to the pox viruses and phycodnaviruses, but has different virion structure from pox viruses and several other properties that distinguish it from the latter. Group: dsDNA. The virins consist of an envelope, a capsid, a core, and a nucleoprotein complex. They are spherical and measure 175 to 15 nanometers in diameter. The capsid is icosahedral with a diameter of 170 to 191 nm and appear hexagonal in outline. The capsomers measure 13 nm in diameter, and there are 1890 to 2172 of these per capsid. Virology. The genome is linear double-stranded DNA and between 170 and 190 kilobases in length. Its guanine and cytosine content is 39%. The virus can be grown in cell culture, but cytopathic effects may be absent. Acidophilic, intracytoplasmic inclusion bodies may be visible in biopsies or post-mortem material. Replication is cytoplasmic and the virins mature by budding from plasma membrane. Life cycle. The viruses in this family infect domestic pigs and their relatives. The natural hosts are warthogs, bush pigs, and argosid ticks. Young warthogs, when infected, develop a high viremia and are infectious to ticks. Older warthogs are generally immune to infection. The virus can replicate within the ticks. Transstadial, transovarial and sexual spread within the ticks occurs. During infection, the virions multiply in erythrocytes, endothelial cells, and leukocytes, and not in the epithelial cells. During infection, the virus can be isolated from blood, spleen, visceral lymph nodes, and tonsils. Several species are known, none of which are able to infect humans. Epidemiology. Viral replication is cytoplasmic. Entry into the host cell is achieved by attachment of the viral proteins to host receptors, which mediates endocytosis. Replication follows the DNA strand displacement model. DNA templated transcription is the method of transcription. The virus exits the host cell by microtubular outwards viral transport. Pigs, warthogs, and bush pigs serve as the natural host. The virus is transmitted via a vector. Transmission route: vector. This family was originally confined to Africa but has since spread to southern Europe and the Caribbean islands.